Hello, guys, welcome to my YouTube channel of physiology. Today, our topic is membrane potential. I hope you will find it helpful for you. It is one of the basic topics of physiology. As we know, there are two categories of cells: excitable cells and non-excitable cells. Non-excitable cells in nervous system are oligodendrocytes, Schwann cells, astrocytes, that are actually the glial cells. They are non-excitable cells. But the excitable cells like skeletal muscle cells, smooth muscles, cardiac cells, neurons, they are all excitable cells, and they possess some sort of membrane potential. Membrane potential is the separation of unequal opposite charges across the cell membrane, or difference in relative number of opposite charges across the cell membrane. Actually, in extracellular fluid, there are so many opposite charges, but they are balanced one. Similarly, inside the cell, inside the cell, that is intracellular fluid, they also have many charges, but they are balanced ones. So the charges that remain unbalanced are involved in membrane potential. Let's see in this diagram, in which there are 5 positive and 5 negative inside the cells, and 5 positive and 5 negative outside the cell. They are balanced ones. So this membrane is unpolarized membrane. There are no unequal, unbalanced charges across the cell membrane. Now let's see in next diagram. There are 10 positive and 5 negative outside the cell. They are the charges. 5 negative would balance the 5 positive. Remaining 5 positive would come around the cell membrane. And similarly, inside the cell, 5 positive would balance the 5 negative, and remaining 5 negative inside the cell would come close towards the cell membrane, and unbalanced outside and inside, they get separated, and there is a sort of attractive force between them, because we know opposite charges attract one another and similar charges repel. So these are the 5 positive and 5 negative that remained unbalanced. They are responsible for membrane potential. As already I have told you that the total concentration of charges is far greater than the charges that remain unbalanced. So the great majority of charges are neutralized by the opposite charges inside the cell membrane and outside the cell membrane. They do not contribute to the membrane potential. Balanced charges do not contribute to the membrane potential. Unbalanced charges are responsible for membrane potential. The most common ions or charged substances in extracellular fluid are sodium and chloride. And similarly, inside the cell, in intracellular fluid, potassium and proteins are the major potassium and proteins are the major charged substances. Although on both sides, sodium, potassium, chloride, calcium, phosphates are present, but protein is an exception. Amino acids get into the cells where they polymerize to form proteins, and proteins are mostly negatively charged, and they get trapped inside the cell, that is in intracellular fluid. Now, what is resting membrane potential? Actually, it is the membrane potential when cell is not being stimulated. Cell is at rest. There is no disturbing stimulus and there is no net movement of charges across the cell membrane. At this time, membrane potential is called as resting membrane potential. As an example, resting membrane potential of neurons or nerve cells is minus 70 millivolt. It is very low potential difference, minus 70 millivolt, because 1 volt is equal to 1000 millivolts, or 1 millivolt is 1 over 1000 volts. The voltage or potential difference that is created across the membranes is very low. The number always indicates the magnitude, and the sign that is present with the magnitude represents the polarity of the inside, and it is a sort of convention. For example, if we say plus 70 millivolt, it means inside is positive, and if this value is minus 70 millivolts, it means potential difference is of 70 millivolts, and negative sign indicates it is the polarity of inside. It is a sort of convention. Why membranes get polarized, or they are electrically charged? Why membranes develop membrane potential? Actually, it is different. It is due to the difference of different concentrations of same ions on both sides of the membrane, and also due to the permeability differences, and also proteins get trapped inside the cell. And one other factor is role of sodium potassium adipase pump. With the help of ATP, with the help of energy, sodium and potassium are moved against the concentration gradients across the cell membrane. Now, this table shows the concentration and permeability relationships between these different charged substances. Sodium concentration is greater in extracellular fluid as compared to outside, and unit is millimoles per liter. Similarly, potassium is lesser in concentration in extracellular fluid and greater in concentration in intracellular fluid. And there are no proteins in extracellular fluid, and proteins are trapped inside the cell and they are present in intracellular fluid. Now, compare the permeabilities. Potassium is 70 to 75 times more permeable as compared to sodium. Simply, you can say if one sodium is crossing the cell membrane, for that sodium, uh, 70 to 75 potassiums are crossing the cell membrane. Simply, permeability of cell membrane for potassium ions is far greater as compared to sodium ions, and this is due to the fact that potassium leak channels are greater in concentration in cell membrane. Now, let's see what is the role of sodium potassium ATPase pump. It is actually the protein that is embedded in the cell membranes, and when ATP is hydrolyzed to ADP, energy is released, and this energy transport sodium and potassium across the cell membrane. For every 3 sodium that goes out, 2 potassium moves in. 3 sodium means 3 positives are going out, and for 3 positive charges that are going out, only 2 positive charges are coming in. And increase in internal negativity occurs because one less positive is coming in, and it increases the internal negativity. One less positive is not neutralizing the anions that are the proteins represented by A negative, because one more positive is moving outside. I have explained it already that nerve cells or neuron cell membrane have more leak channels of potassium, and concentration gradient of potassium favors it to move outside. Role of sodium and potassium ATPase pump. Sodium potassium ATPase pump is responsible for just 20%. It contributes 20% for the establishment of minus 70 millivolts, because for every 3 sodium, for every 3 positives that goes out, only 2 positive comes in. Hence, outside becomes more positive and inside is getting more negative. Why potassium is more inside the cell as compared to outside? Simply, you should remember that 10% of the potassium is present in blood plasma and 90% of the potassium is present inside these cells, that is in intracellular fluid. This is due to the hydration shell, a sphere or shell of water molecules that surround these ions. The shell or sphere that is formed outside the potassium is smaller as compared to sodium, and these ions have to come out of the sphere of water molecules. Shell of these ions have to come out of the shell of water molecules to combine with other substances. Due to the smaller size shell of potassium easily moves out of the shell and then and binds with proteins. Therefore, potassium is mostly present inside the cells. Now, here we will discuss the individual roles of sodium and potassium in generation of resting membrane potentials. First, we will discuss the potassium role alone. And it is an hypothetical model, because inside these cells, both ions are simultaneously involved. It is just an hypothetical or experimental model. How can potassium affects the membrane potential? As we know, and in table we have shown that potassium concentration is far greater inside the cell as compared to outside. Therefore, concentration gradient favors its movement out of the cell. But when potassium moves outside, anions, proteins present inside the cell, they have negative charge. When positive charges are going out, negative charges are increasing. Already outside is more electropositive and more potassium ions are coming out. Similar charges repel each other. So a sort of electrical gradient develops outside the cell that pushes the positive potassiums to move from outside to the inside. When concentration gradient towards the outside and electrical gradient towards the inside counterbalances each other and becomes equal, then there is no net movement of potassium. Net movement of potassium means if two potassiums are moving out and two potassiums are moving inside the cell, there is no net movement. So such a potential at which there is no net inside or outside movement of ion, such a potential is called as equilibrium potential. Equilibrium potential of potassium is minus 90 millivolt. At minus 90 millivolt, there is no net inside or outside movement of potassium. Similarly, now we will discuss the role of sodium alone. We know sodium is far in greater concentration outside the cell as compared to inside. So concentration gradient is favoring the net inward movement of sodium. When sodium moves to the inside, uh, positive charge gets accumulated inside these cells, and sodium is moving inward. In extracellular fluid, most common anions are chloride ions. When sodium are moving inside, they are taking with themselves the positive charge, and negative charges are increasing on outside. Outside is already positively charged. When net inward movement occurs, more positive charges are on the inside. So electrical gradient for sodium is from inside towards outside, because similar charges repel each other. When both concentration gradient and electrical gradient cancels their effect. Such a potential is called as equilibrium potential, and equilibrium potential for sodium is plus 60 millivolts. Plus means the polarity of inside. The point to remember here is that sodium tends to drive the membrane potential towards its own equilibrium potential, and similarly potassium tends to drive the membrane potential towards its equilibrium potential. And equilibrium potential is mathematically expressed by Nernst equation. One thing another that I'm going to repeat again, these are the hypothetical models, role of sodium alone and role of potassium alone. They are hypothetical model, and they help us to study the concurrent effect, because in cells both the sodium and potassium act together at the same time simultaneously. Potassium concentration is greater inside the cell, so concentration gradient is towards the outside. But when potassium moves towards outside, internal negativity due to proteins is increasing. Similarly, sodium concentration gradient is towards inside, and when sodium are moving inside, outside positivity is decreasing. But we know potassium permeability is far greater than sodium. Therefore, more positive charges are coming outside as compared to the positive charges that are coming inside. Hence, outside becomes more positive and inside becomes negative. Potassium's tries to bring the membrane potential towards its equilibrium potential, but the membrane potential is slightly less, that is minus 70 millivolts. Membrane potential is not minus 90 millivolts. This is due to the fact that some of the sodiums that comes inside along with their positive charge, they neutralize these negative anions. Therefore, the membrane potential is minus 70, not the minus 90 millivolts. This is the Nernst equation, in which 61 is a constant. Z represents the charge of the ion, for which you are going to measure the equilibrium potential. And CO is the concentration of an ion outside the cell, that is in extracellular fluid, and Ci is the concentration of ion inside the cell. So this mathematical representation is general formula for any of the ion, uh, any of the ions equilibrium potential. So we can, if we know the concentration of an ion in extracellular fluid and intracellular fluid, by using Nernst equation, we can find its equilibrium potential. I hope it has helped you out. If you like it, please share and subscribe my channel. Thanks a lot.